Our next speaker is in the host pathogen arena is Peter Williamson. Peter is an MD, PhD, a physician scientist interested in host pathogen interactions, focusing on the fungus Cryptococcus. Currently, he is at the National Institute of Health in Bethesda. His laboratory functions on mechanisms that control fungal virulence as well as host responses and susceptibilities in human patients and using animal models. Current projects concerning the fungus focus on post-transcriptional regulation of RNA networks and molecular evolution of the fungal infectome. Studies of the host response include the role of messenger RNA dependent regulation of adaptive and innate immunity, as well as the roles of immune dysregulation and genetic defects in human disease susceptibility. So Peter is going to talk about the host side of this organism.
I'd like to thank everyone for inviting me to come and give a talk here. I'm going to talk a little bit, I'm going to focus down. We're going to look at the host and the issues involved with our patients here. So this is, I've just recently moved from Chicago to the National Institute of Health in Bethesda, Maryland. And let's see how we can get this pointer to work here. There we go, very good. So the nice thing about NIH, and the reason why we came, was to really figure out how a lot of these things, these basic science approaches, I came from a pathogen side, how we could apply this to the human patients. And so the nice thing about NIH is we have a nice hospital here, and my lab is right back down here. So all I have to do is run down, and I can see my patients, and I can deliver specimens, and we can do a lot of good science to try to figure out why our patients are not doing so well. So, let's see, there we go. Is that advancing? There we go. So we've been focusing on non-HIV cryptococcal meningitis. With the advent of antiretroviral therapy, you can see that HIV-related cryptococcal hospitalization and deaths have declined, although not remarkably. You know, it's amazing. You'd think it'd be wiped out by now, but really the incidence of Cryptococcus, the hospitalizations, has only gone down by about half. And I think this is reflected overseas as well. But the non-HIV cryptococcal infections, you can see, have been quite stable, that there really have not been any decline during these infections. Not advancing well. There we go. Oh, back. There we go. So Cryptococcus is everyone, I think, there's a very large contingent of physicians I can see in the audience. You're quite familiar with cryptococcal disease. And we know about the terrible outcome that exists here. AIDS-related, we're seeing 15% mortality, up to 50% in developing countries. Non-HIV, 10 to 30% mortality. So it's still a bad disease, and there's still a lot of controversies on really how to address this. This is from the latest cryptococcal guidelines. In refractory disease, for instance, adjuvant immunotherapy with recombinant interferon be considered a refractory infection with a concomitant use of specific antifungal drugs. I think in the non-HIV infections, the paradigm has been from HIV. We've done a lot of studies with HIV. We know a lot about the immune system and how T cells are suppressed. And I think a lot of those paradigms have been applied to the non-HIV population. So we really started our group, started our study looking at a series of non-HIV cryptococcal. These are previously healthy patients. So of those 3,000 a year that we still seen in the United States, probably about 1,000 or 1,500 of those are in patients without any previous infection. And I think we've all seen these. And so we have a protocol where we follow patients with meningoencephalitis. We also have local disease like bone and pulmonary disease. And we also have a cast of normal volunteers who voluntarily submit to lumbar punctures as our control, which is quite a feat. And we have an acute phase where we study, and then we have a resolution phase. And this study really started to look at some of the genetic susceptibilities and the immunologic susceptibilities behind these infections. But as we started to recruit these patients, we found out they were really sick. I mean, with a 30% mortality, you can imagine these are pretty sick. And so we're bringing in a lot of patients, and a lot of the clinical challenges there have been really quite impressive. And to do this, we've been able to assemble quite a team here we have, oh boy, this is really funny. There we go. Of clinicians, led by our staff clinician, Neil Panicol, with neurology and neurosurgery, and our person who's helping. She's a multiple sclerosis person who's been developing a lot of standard methods for doing a lot of immunophenotyping in the CNS. And then our Dima Hamoud, who's been doing a lot of our immunoimaging as part of our study. And this has really made this very possible. And so, for instance, Prashant Chittabuena, he rides in on his bicycle during the 2-foot snowstorm to see our patients. He's putting in shunts. It's a very active team for a lot of very, very sick patients. Most of these require shunting, a lot of infection. So as we began to recruit these patients, this is our first patient we had. And it was a 58-year-old male, presented in a usual way with dizziness, hearing loss. We don't see a lot of fevers in these patients. CSF cultures grew Cryptococcus, was treated with standard therapy, amphotericin, then switched over to fluconazole, and then he worsened. I think a lot of times the clinicians, we would often think that you would get about, in non-HIV, we give about 4 to 6 weeks of amphotericin, as Dr. Bennett talked about yesterday. And then as we switch over to fluconazole, often that's about the time they worsen. And we always interpreted that as a failure of fluconazole, because as Arturo was mentioning yesterday, I think we're beginning to become more aware of the host. But our paradigm is more always centered on the pathogen, trying to kill the pathogen. Every problem is interpreted in terms of killing the pathogen. If the patient's not responding, we're not killing the pathogen properly. And so this was really a challenge for us. But this patient was failing therapy, was brought over to NIH, had a little bit of parenchymal leptomeningeal enhancement on his flare intensity. And you can see on the Glasgow coma score here, which if you do a 10-day moving average, you can see it was around 13. So that's a pretty obtunded patient. And you can see the time frame here was really a long time. And we went along, we were adding all these antifungals, trying to kill the fungus all the way along. The cultures were negative, actually. But the reason why we don't believe them is Dr. Bennett had originally shown way back when, before we had fluconazole, when we treated with amphotericin, there were about, with a culture negative in the CSF, about 15% of those recurred. And so we began to distrust those cultures. And I think we still should, but I think with fluconazole, it does give us a little bit more of a degree of safety there. But here we go. We were going along in here for quite a long time, and then we decided to use those guidelines. We gave gamma interferon. You can see he dropped. He went into a deeper coma. So we said, we can't do this. Let's give him steroids. And so we gave him a bolus of steroids, and he woke up in 3 days. And you can see the Glasgow coma came up to normal, and the antigen went down too, which was even more remarkable. We hadn't changed the antifungals during this time, but the patient really woke up. And we had another patient came in, and this was pretty remarkable here. I got a call right about here. The patient was at an outside hospital, and he'd be admitted to a neurosurgical service, and he was diagnosed with cryptococcal meningitis. Well, we all know neurosurgeons put everybody on steroids, right? And so she was going along very well with a gaudy eye infection, and a lot of inflammation, cryptococcoma, getting better here. And then they called me about this stage. And what had happened was, the first question I asked, this was about after the 3rd patient we had started to give steroids on. I said, when did you stop the steroids? And there was a big pause, because that was one of the first questions I asked. And he actually felt guilty because the patient had been on steroids. But actually what happened was he was on the neurosurgical service on prednisone, and then he got transferred to medicine. Whoa, we don't use steroids. Stop the steroids. Worst thing in the world. And you can see here his lesions inclined. And the inflammation increased. This is on a flare image down here where you can see the inflammation. And the inflammation increased, and the patient went back into a coma, was back in the ICU. So I said, start the steroids. And when he's better, put him on a plane and send him to NIH. So we bumped up the steroids, 50 milliequivalents, 50 of prednisone, which is not a huge dose. And you can see on the same therapy of amazome all the way along here, it starts to increase. Then we get him at NIH and we go, well, this has been quite a long time. This is over a year, you can see. So after about 2 months here, you go, well, we ought to be able to taper this down. So we tapered it down to 25. And then everything worsened again. And in fact, I've still got the emails where they were sending their emails back to NIH saying, back to their physician in Seattle and saying, oh, what are these NIH people doing? They're cutting the steroids and her mental status is going backhill. They're going to go to the ICU. Well, instead, we popped back up on the steroids and the inflammation went down and we were able to just taper her over a period of time. And now she's back at her job. She has completely normal mental status. She works as an accountant and she's able to do all her job and doing well. But you can see this took a long time, but how important these steroids were. So we're like, what does this mean? You know, I mean, they have this response. There's this controversy in the literature, gamma interferon, steroids. We need to get some objective data. So in mice, okay, we have quite a nice model in mice, and we can infect mice orally, or we can infect them by a posterior pharyngeal route. We can get lung infections. We can show dissemination of the brain. Really nice work that has been done over the years. And you can see here that Gary Hufnagel a number of years ago showed CD4, CD8 cells were very important in cryptococcal infections. We can show that MyD88 and TOL2 is important as well, and TOL9. So we can get some basic immunology. But mice also have some limitations, okay? So here's two different mice, C57 and SJLJ. And you can see that if you infect the mice to a posterior pharyngeal pulmonary model, you can see that one mouse, the C57, the CFU in the lung go up quite a bit, whereas the other mouse will clear it. And the number of cells that you recruit in the two different mice. You can see with the C57, we see things like eosinophils, which we don't tend to see in cryptococcal infections. So it's not a very good model of human infection. When's the last time you saw an eosinophil in a cryptococcal infection in humans? And yet C57, and we use them all the time too, because a lot of our knockouts are done in that, they're producing a lot of cells and a lot of polys, a lot of eosinophils that we just don't see in human infections. In the resistant mice, it behaves a little bit more like the human infection, but you can see there's a deficiency in the model systems here. They're very good for mechanisms. So we do knockouts. We have a knockout mouse that we're about to publish shortly, where you can do a lot of cool mechanistic biology, but it also has to be informed by what's going on with the human. And that's very important. So we decided to look at some of our patients and to really get some objective data. And for that, we looked at 17 consecutive CNS cases. We looked at 6 non-CNS cases, and none of these have any underlying risk factors. And that allows us that we don't have to worry about if it's a transplant patient, what's going on with the immunosuppression, all that kind of thing. Just allows us to be able to stratify this patient population a little bit more carefully. A lot of mental status changes going on. Actually, all of them all had mental status changes of various amounts. The non-CNS group was a very nice control group because we're always worried if our control group was exposed to Cryptococcus. And clearly, a patient with pulmonary Cryptococcus has been exposed to Cryptococcus. So for some of our immunologic tests, that was really important. And we had a number of median age of about 54. It's kind of an older group. And it was a sort of a variety between Gaudii and neoformans. I expected to see more of a difference clinically between neoformans and Gaudii. But it takes large numbers to be able to see that. CD4 count in this group was fairly normal. Okay, so this is not a group of idiopathic CD4 lymphopenia. And the white cell count was the usual 30 or so, and the cryptococcal antigen was lowish as usual. In HIV, you have much higher cryptococcal antigens. And most of these patients were not on prednisone at the time when we were studying these patients. So this is a sort of simplified scheme here. I'm going to use the dendritic cell and the effector T cells, which recognize the activated dendritic cell, gets activated by the Cryptococcus through a number of toll receptors, mannan receptors, and beta-glucan receptors. The T cells, dendritic cells synapse after activation, produces TNF-alpha and gamma interferon, which activates macrophage and other phagocytic cells with participation of other tissue factors like GM-CSF, and in an effective response produces iNOS, phagocytoses of the cell, and this is called an M1 or a classically activated macrophage, and this would be the effective response, okay? And this macrophage will be producing TNF-alpha and IL-12. TNF-alpha is a major reason why we get fevers. And you notice in Cryptococcus we don't get many fevers in the non-HIV infections, which is kind of a clinical clue to what's going on here. And so first looking at the dendritic cell T cell effector, we wanted to systematically go through this synapse and see what in the heck are these patients doing. If we don't know what these patients are doing, we don't know what to do as far as therapy. So it's going to help us provide some biomarkers, some theoretical understanding, so we know where to go to treat these infections. So what we did first is we got peripheral monocytes from patients, and we produced immature dendritic cells by a cocktail which sort of expands these. And then in the presence of either manoprotein or cryptococcal lysate, we were able to then activate these dendritic cells. And then we co-incubated them with CD4 cells from either the CNS patients or our non-CNS control patients. And we used blood and CNS and blood and CSF. And we exposed them to this, co-cultured it, and then we looked at things like proliferation and production of Th1 cytokines like gamma interferon, TNF-alpha, GM-CSF, IL-17, to be able to see, was this synapse important? If there was a dendritic cell defect, we'd be able to see, or a T cell synapse defect, we would see a defect in these patients. And what we found, we expected to see something down. But instead, look at this. Here's an example of one of the patients. From the CSF, we have a control here. This is gamma interferon, this TNF-alpha production. You can see in the presence of the manoprotein or cryptococcal extract, they're producing huge amounts of TNF-alpha, okay? These patients' T cells are working like gangbusters, all right? So putting gamma interferon in a patient like this is like putting gasoline on a fire. You're not going to be improving anything. In fact, you might be killing them. Now, this assumes that the culture is negative at this point, okay? So I always want to put that on the back of your mind. The interesting thing was in blood cells, T cells from the blood, almost nothing. So this is a very compartmentalized infection. So if you want to study the meningitis from blood, forget it, because the action isn't going on there. CRPs aren't very elevated in these patients. It's all going on in the brain. Conversely, your immunosuppressants that are working really well systemically may not be working so well in the CNS. So this is just the summary statistics showing that in the patients with the meningitis had huge amounts of gamma interferon production by CD8 cells, and this came out in CD4 cells as well, and either with the manoprotein or with crypto extract. Okay, so you get this activation that's going on. And so the next thing is look at the cell types. Ooh, I think this was supposed to be a cell. Well, that was supposed to be the schematic again. The next thing we wanted to look at was the T cell activation in situ from the CSF. And so with Bibi Belikova, she had worked out these very nice protocols for looking at, because the problem with CSF, you don't have many cells. So you have to have one tube that you can do, and you have to get everything you can out of that one tube. So Bibi has worked out some really nice ways of looking at activation parameters that can use for both T cells and can be used for B cells and can be used for NK cells, and you can reuse this a lot. And we developed, and with a 12-color flow, you can get a very nice panel and look at the cellular response here. And what we found was this. I'm not going to go through all of those cells. That would put you all to sleep. But just looking at some examples, HLA-DR is a marker of activation of CD8 cells. And what we saw here was that in the CSF of patients with the CNS disease, they had a markedly increased activation of their CD8 cells, CD4 cells as well. The non-CNS, of course, they didn't in the CSF. They didn't have much activation going on, and that was our control group. NK cells were activated as well. The monocytes were activated as well. And so this is a very activated milieu. So again, coming in with using that paradigm from HIV on this non-HIV cohort is going to get you in trouble. It's all about the host. Very important. We looked at soluble markers in the CSF. Once again, you can see gamma interferon production off the wall, okay, compared to here. This is a log scale, so it's over 20 times normal. Gamma interferon responsive chemokines like CXCL10 elevated as well. Pro-inflammatory cytokines like IL-6 up as well. IL-10, which is a macrophage activation marker, as well was up. But it's a little flaky because IL-10 doesn't necessarily signal that macrophages are appropriately activated, as I'll get into here. Now, the action is going on in the brain. You know, when we tap a CSF, we go 30 cells. How could that be doing anything? When we get bacterial meningitis, we get 10,000 cells. That looks like a really angry inflammatory response. Cryptococcus, 30 cells. What could that be doing? No big deal, right? but the action is going on in the tissue. When we do biopsies, when we look at autopsies of these patients, here's the meninges. You can see the anti-CD68, which is the macrophages. If you look at the Virchow-Robin space that penetrates into the brain down each vessel, you can see a tremendous monocyte infiltration that's going on here. You can see a T-cell infiltration as well. So this inflammation is going on in the brain, causing damage. Now, is it causing damage? Okay, so the next question. Because you'd say, little inflammation. Well, that's a good thing, right? Your leg swells up, it goes down, your leg's okay, okay? But the brain is a compacted space. You've got this brain, this skull inside of you. You can't really tolerate a lot of inflammation. So this is kind of a cool marker. This neurofilament light chain, we use it as a marker. It's an axonal protein, and it's released into the CSF during damage. And this is kind of a cool study that was done. It's been used in multiple sclerosis. It's been used in a lot of inflammatory brain diseases. And this was kind of a cool study. This guy had gotten knocked out in the ring. And somehow they did spinal taps on him like every day, just kept doing it. And when they did that, they showed, I'm not so sure of that. When they did that, they showed that the neurofilament light chain was up around 3,000, normal's around 30 or so. And as this guy recovered from his post-traumatic stress syndrome, it improved and it finally went down to normal. So it's not an acute phase reactant necessarily, it's actually released during axonal damage. And what we found out was, here's one of our patients, here's soluble CD27, which is a marker of T-cell activation, and you can see a linear association there between the inflammation that's going on here and the neurologic damage. And by the way, his highest level was not 3,000, but 20,000. So this is during a cryptococcal meningitis. It's like being knocked out 10 times. So that inflammation that you're seeing, that you're saying, oh, that's good, it's killing the Cryptococcus, it's also killing the guy's brain. So when a year or two out, you see this guy sitting in front of the TV just kind of wandering things, it's because of all this neurologic damage that's been going on there. We're finding in some of our neuropsych testing, they're down around the 10% range of where their mental state should be for an equivalent control. So this is not a benign event, okay? And just our summary statistics, neurofilament light chain, is a good 1,000 or so higher than it normally should. So this is a very, very damaging kind of an environment. So let's finally look at the final part of the macrophage activation here. And M1 activation is really what you want to be able to kill this fungus. And so we expected with this excitable T-cell environment, the macrophages should be activated here as well. So we looked at, here's a biopsy of a patient, and we looked at autopsy patients as well. And again, in the meninges, you have the CD68, the CD163, which are macrophage markers, CD3, a lot of cellular inflammation inside the brain. But when we look at activation markers for the macrophage, INOS being the M1, the activated one that's going to kill the Cryptococcus, nowhere to be seen. What's going on with these macrophages? When we do an M2 marker, which is a marker of alternative macrophage activation, which is not an effective immune response, you can see these cells are lighting up like crazy. And if we stain with calcifluor white, just to stain the fungus, and then we look at the macrophages, we notice that the macrophages in the crypto are not in the same ballpark. The macrophages are just sitting on the beach, just having a good time. And the T-cell, fire is going on, causing all this damage, but the macrophages are just hanging out, not doing anything. And so this was very important to relate to our clinical response, because remember, those steroids, they were quite effective, but they had to be kept on for a year. And the reason is because these macrophages are not clearing that antigen. The fungus might be dead, but that antigen is still there. When you do an LP and a year later, off therapy, you can still see beautiful fungi. They don't grow, but they're still there. You do a biopsy, the fungus is there. Everybody's worried. Therapy 2 months ago, the patient's deteriorating, the cultures are negative. I have to put on more antifungals. You wouldn't think that way. So I'm trying to bring that over to the non-HIV cryptococcal infections and think the same way. Always think in terms of the microbiology, thinks about the immunology. The problem is we don't have good biomarkers at this point. So what we're developing now is to try to work at getting those biomarkers in order to be able to understand this disease. I think, let's try, see where I get my first slide. I don't know, I'm kind of advancing, but nothing's happening here. Can you give me the first slide that has something on it? Okay, we'll try to go from there. Okay, so we wanted to figure out the susceptibility. Why are these patients getting infected? And what we did find was that a number of these patients, I didn't show you the next case, which was a dancer. She was a 20-year-old dancer. Doing fine. Had cryptococcal meningitis. She had actually gaudy eye infection. She was from Northern California. And about a year later, we brought her in from NIH as part of our study. And we did a CAT scan on her. She had a very impressive infiltrate. But it wasn't a cryptococcal infiltrate. You know, with Cryptococcus, you get that very dense kind of core lesion. This one had a diffuse ground glass across the CNS, and again, across the CT. And actually, it showed what's called crazy paving, which is this alveolar thickening. And that's typical. I brought it down. I said, what in the heck is this? I couldn't remember. I brought to our radiologist, said, well, this is alveolar proteinosis. And I was like, what? What's that? So in going over and looking at that, we found out that these patients produce an antibody against GM-CSF, which is the activator, granulocyte monocytic cytokine soluble syndrome, which activates macrophages. It's part of that tissue environment. If I dared go back, I could show you the little GM-CSF on the macrophage, but I won't attempt to do that. And we found was that the anti-GM-CSF antibody reacted from the patient against recombinant protein. And more importantly, because a lot of autoantibodies don't have function, this work was done by Sarah Brown at NIH. She showed that the antibody would block STAT5 phosphorylation, which is a marker for activation here as well. That the IgG fraction in the patient would block the activation here of the phosphorylated STAT5, and the plasma overflow would not, showing that this was an autoantibody. And basically what these patients are doing is GM-CSF normally activates through this pathway, and actually we're studying this now and showing that it works through autophagy, and then produces IL-1 beta, which activates the macrophages in a feedback mechanism. And what happens is, in addition, this syndrome typically causes this infiltrate in the lung because the macrophages can't clear the protein that's normally produced in the lung. And in the same way, it's not able to take up Cryptococcus, and it's not able to process and kill that. And so we're finding out that these patients, for the most part, don't have a T cell defect, but they have a macrophage defect. And let's see. And so pulmonary alveolar proteinosis was first identified back in 1958 by Samuel Rosen. And actually the second person is Castleman, that you always probably know from Castlemans Disease. And this is a syndrome that occurs in about, in about 5% of our NIH cohort, and mostly with gaudy eye infections, as was shown later by Kwang Chung's group et al. But what happens is that there's a number of congenital reasons. You can have hematologic malignancies, you can have occupational exposures. There are a number of, some of the GATA2 infections, there's been one association with cryptococcal meningitis. But most of them are due to this neutralizing anti-GM-CSF autoantibody. And as was shown years ago, that the GM-CSF antibody, if you put it into a macaque, you can reproduce this disease. So it's an important but small cause of this syndrome. But it's been a nice model because, remember, the other patients showed a macrophage defect. And what we're finding out is genetically, I'm not going to go into this disease, this data, but a number of these patients show genetic defects in their macrophages as well. And so you can have extrinsic defects from the GM-CSF autoantibody, and you can have intrinsic defects from genetic defects in that signaling pathway involving macrophages. But it does bring to mind what Arturo was talking about last night, which was the host damage hypothesis. So in cryptococcal HIV, we have an iris-like reaction. And here's this pathway again with dendritic cells, T cell effectors, and macrophages. And when you treat a patient with HIV with antiretroviral therapy, all of his immune system comes back. It might come back slowly and irregularly, but it's all there. And so when you have activation there, you have, this is sort of the inflammation as well. So you have this tremendous amount of inflammation that's going on here, and you get the typical iris reaction. The thing is, the pathogen clearance is also fairly intact. So this whole thing with iris goes off in about a month or so. In fact, Irene Soretti, who studies iris in our institution, says, how come my patients are out of here in a month, but your patients are in here for like 6 or 8 months? And the reason is because with the HIV iris, you have this pathogen clearance, which comes back, and that clearance occurs fairly quickly. It's still a very horrible disease and needs some immune suppression, but you have that response. In another disease like TB meningitis, you have sort of a halfway response. The T cells are sort of halfway activated. They're plus or minus. So they produce IL-6 and gamma interferon, but they also produce IL-13, so they're sort of halfway in between. And their macrophages are sort of halfway in between. So they have a fair amount of inflammation, but their pathogen clearance is kind of reduced, and the inflammation is a little bit reduced, but it's still enough inflammation that steroids are beneficial in this case. Now, in our case, the cryptococcal non-HIV, we have the worst of both worlds. We have all the badness of all the T cell activation, but we don't have the macrophage clearance. So what we have is a macrophage T cell dissociation. We have all the inflammation that's going on from the T cells that cause that damage, but we don't have that macrophage clearance. So therefore, it takes a long time to clear that antigen. So that's why you see those organisms in the CSF, you see them in the brain long after they're dead, okay, because you can't clear this infection. Okay. So in summary, what's we've been using NIH for is sort of an incubator. You know, we can do things that's very difficult to do on the outside. I came from Chicago about 5 years ago. You always had insurance companies, you know, send the patient home, right? And so it's very difficult to keep the patient in for any length of time. Whereas we can keep the patient in, we can do these kind of therapies, and we can follow them very closely by sequential LPs and following them very closely. Things would be very difficult. And so we look at our kind of institution as being able to develop some of these protocols, and then in the larger series, we have a UO1 with Karen Marr at Johns Hopkins. Many of you might be in this part of this cohort already, where we can begin to use some of the biomarkers that we've identified and try to do more larger studies to validate these, and in different populations as well. The transplant population, as you know, is a whole other bag of beans and can be kind of a variable response, somewhere between HIV and the non-HIV cryptococcal, the previously healthy patients, and you can have something in between, and those are even more challenging in many ways. So hopefully we can go forward with this, develop some biomarkers so that we can have some way to dissociate that microbiologic and inflammatory response individually, so that at least in resource places where we have more resources, we'll be able to treat them individually and be able to get patients through this immune response as well. Okay, thank you very much.